Good evening. Good evening, my dear students. Today we discuss about the biochemical identification test. This test basically the identification test for the microorganisms. The biochemical procedure by which you can identify the microorganisms. Last class we discussed about the IMVIC test or indole methyl vinyl vascular and cited utilization test. This test for the identification of the basically this microorganism produce some specific biochemical parameter, and those parameter can identify by the different specific different reagents, and ultimately we are getting the which organism produce from this. The first test is the indole test or I. This basic principle to determine the ability of the organism to split indole from the tryptophan molecule. Those organisms which produce tryptophan is the indole group of amino acid which produce with some specific organisms, and organism produce tryptophan, and tryptophan can be identified by the specific reagent. The indole is one of the most metabolic degradation product of the amino acid of tryptophan. Bacteria thus process the enzyme tryptophanase and is capable to hydrolyzing and determine the tryptophan with the production of indole, pyruvic acid and ammonia after deamination procedure. Indole, the media is reagent for this process. The tryptophan 1% or the peptone broth. Basically, tryptophan is an amino acid or peptone broth is a protein. Protein, that means the amino acid. For the growth of the bacteria, amino acid is needed. The reagent, one reagent the Ehrlichia, another is the Kovacs reagents. Ehrlichia reagent contain para-dimethyl aminobenzaldehyde and ethyl alcohol and hydrochloric acid. Kovacs reagent, it is also used the para-dimethyl aminobenzaldehyde and pure amyl and isoamyl alcohol and HCl. Indole other media which can be used for the indole production: sulfite indole moiety agar media and motility indole ornithine agar media. The two solid media can use to identify the tryptophan producing microorganism, but in that case the test will be positive in case of E. coli because E. coli produce tryptophan, and tryptophan react with the Kovacs reagent, they produce the color, and negative in case of Klebsiella pneumonia. This is the test procedure by which you can identify the tryptophan producing microorganism. Inoculate peptone broth with a test organism and incubate for 18 to 24 hours at 35 degrees centigrade. Add Kovacs reagent contain alcohol to peptone, water and extract to insoluble upwards. Indole para-dimethyl benzaldehyde quinidal compound, red compound is formed in presence of tryptophan. To interpretation and development of the red violet color at adding the reagent in the indicative of the presence of the indole and its positive test. But those organism which not produce any tryptophan containing amino acid, this test will be negative, like Klebsiella pneumonia. Indole rapid test. Apart from these two tests, the indole rapid test can be done by the indole spot test by the filter paper. In the filter paper, the microorganism is placed here. In that you use the Kovacs reagent, ultimately the color will be changes. The indole spot test for the anaerobic bacteria, indole micro technique. This is the very sophisticated technique by which you can identify the microorganism. A single separated microorganism placed here. The indole strip are used, contain the reagents, ultimately color will be produced. Those organisms which produce the indole test in case of positive, in case of number 1 E. coli, Proteus vargadis, Klebsiella oxytoca, Klebsiella ornithoca. So these are the bacteria which are the positive test, and the negative in case of Salmonella, Klebsiella Enterobacteria and Proteus in malariasis. Another second most test by which you can identify the microorganism, the basic principle to test the ability of the microorganism to produce and maintain the stable acid and the products from the glucose fermentation and to overcome the buffering capacity of the system. And this is the qualitative test for the acid production by fermentation. Methyl rate. Methyl rate generally the indicator, chemical indicator which is used for the different method. But here this methyl red is the pH dependent color indicator which is the range between 6, it is produced red color. In case of 4.4 it is red color. The pH at which the methyl red detect acid is considered the lower than the pH of this indicator. Thus, to produce a color changes, test organism must produce a large quantity of acid forming substrate. Ultimately, those organisms which produce acid by the fermentation of glucose, ultimately the methyl red test will be positive. Methyl red positive, they generally the two broth are used. One is the methyl red broth and PV, the polypeptide glucose, dipotassium hydrogen phosphate and distilled water, and MR pH indicator 0.1 L 300 ml of 95% ethanol distilled water 200 ml. In case of positive E. coli, in case of negative Enterobacteria erosions. Methyl red test procedure, you can see it very carefully. The methyl red MVP are birth in the pure culture. The test organism and incubate 35 degrees centigrade for 48 hours to 72 hours. At 5 drops of methyl red reagent, ultimately the red color will be produced. If the pH 4.4, if the pH higher, the 6 will produce yellow color. The positive color will be sufficient acid to allow and reagent to maintain the distinct red color, and the color pH in the 4.4 in the surface of the medium. Negative yellow colors pH 6 point at which the surface will be medium. The methyl red positive in case of E. coli, Neisseria piscis and Listeria monocytogenes. Negative in case of Enterobacteria aerogens, Enterobacter clovans and Klebsiella and gram negative bacteria are the methyl red are the negative. Voges-Procier's test. This is the acetone production. The basic principle to determine the ability of the microorganism to produce a neutral endpoint at the acetyl methyl carbamide or acetone from the glucose fermentation. And in case of it is positive Enterobacter aerogens, negative E. coli just reverse. The Voges-Procier's test, in this case the pH is 6.9, polypeptide glucose, dipotassium phosphate and distilled water. Reagent alpha naphthol 5%, colorimetry and absolute ethanol and 40% potassium hydroxide, oxidizing agent and the distilled water. The basic procedure for the indole pure culture testing the MRVP broth indicates the 24 hours at 37 degrees centigrade. The alicate of 1 ml of broth to a sterile test tube and 0.6 ml of VP and 0.2 ml of snake venom with gently exposed to the medium and atmospheric oxygen. Allow to tube to remain undistributed for 10 to 15 minutes. Interpretation. The positive in case of pinkish red color at the surface of the medium, negative yellowish color at the surface of the medium. So generally the glucose in presence of potassium hydroxide acetone is produced. The acetone with atmospheric oxygen produce diacetyl plus water, and these are diacetyl plus guanidine component of peptone alpha naphthol, pink color will be produced. This is the basic principle, basic chemical reaction of this Voges-Procier's test. This Voges-Procier's test is another alternative test. This is a very sophisticated and expensive test. By the gas liquid chromatography measures by the diacetyl and electron captured gas liquid chromatography and gas chromatography, chemical ionization, mass spectrometry and colorimetry method measurement of the diacetyl. So this is the process. This is a very sophisticated and cost effective, not a very easier technique. The precaution taken in the Voges-Procier's test, the organism which be MRVP and negative or vice versa, The most organism belonging to the Enterobacteria. Organism like the Haphenia and Aphelia and the Postius mirabilis may be given the MR and VP positive result through the VP sector isolate. Excess KOH may marks a weak PV positive reaction. The Voyagers-Pocket test in case of positive in case of right hand is the negative in case of positive positive in case of left-handed. Klebsiella pneumonia in case of negative E. coli and others organisms. So list of some specification of cited utilization technique. This is the last one. This test is used for the determine the ability of an organism to utilize the sodium citrate, and it is only the carbon source and in the inorganic ammonium salt of its southern nitrogen sources, and bacteria that can grow on the medium and burns with the bromophthalin blue indicator for the green to blue. So cited utilization test for the two media, one is the Kosar medium and another Simon cited medium. In case of Kosar cited medium, the reagent is the sodium chloride 5 grams, magnesium sulfate 0.2 grams, ammonium hydrogen phosphate 1 gram, potassium dihydrogen phosphate which is used in the buffering solution, and Kosar medium 1 liter and H agar 20 grams and bromophthalin blue 0.2% 40 ml. And cited utilization test using the sterile technique inoculate the bacterium into the appropriate level Simon's cited tube and means the spot inoculation and inoculation of the center. In case of positive, the Klebsiella pneumonia, negative in case of E. coli. These are the tests. In case of positive, test develop a deep blue color within 24 to 48 hours, including the test organism have been able to utilize the cited present in the medium and which production of the alkaline production. Negative, no change color, generally green color. The Simon cited test, it is the slant culture, it is in the broth culture. It is contain the agar, it's not contain no agar. It contain the bromophthalin blue indicator, here no indicator is used. Positive test is shown. Positive test is indirectly growth and change of the color of the medium and present the turbidity of the medium in case of Kosar cited reagent. In case of positive Klebsiella pneumonia, Cytobacter diversae, Enterobacteria, but in case of negative Escherichia coli or E. coli, Salmonella typhi, Salmonella paratyphi, A. Shigella species, Neisseria, Enterobacteria, and Edwards. So thank you very much for listening your lecture. If you like me, please like me, or if you any question, please question in our chat box or in the Google Classroom. We discuss in the next class. Thank you for your patience to listening the lecture. Thank you again.